Hello and welcome to the Metabolic Classroom. I am Professor Ben Bikman. I'm a biomedical scientist who focuses on metabolism, and I'm a professor of cell biology. There are a lot of things we could discuss when it comes to alcohol, but this is, of course, one of the most active or one of the most commonly consumed psychoactive substances around the world. Ethanol, the main form of alcohol, is psychoactive. It does influence the brain. And this is what we find in all sorts of alcoholic beverages. The metabolism of ethanol or alcohol is unique in that it is metabolized almost exclusively by the liver. And we're going to revisit that fact as we discuss some of alcohol's effects on metabolism and insulin resistance later. Now, as I mentioned, there's a lot we could discuss with regards to alcohol consumption, and we're going to keep it strictly within the realm of metabolism and insulin resistance. And if I sometimes say ethanol or alcohol, this is synonymous. So it's the same thing. I may just shift between the two depending on the particular study that I'm thinking of or citing, or whatever comes to the top of my mind in that moment. Now, alcohol-induced insulin resistance is a thing. Ethanol is capable of causing insulin resistance both directly and indirectly. And there are multiple molecular mechanisms that are responsible for this. So in other words, alcohol is capable of causing insulin resistance actually through multiple and distinct methods or pathways. And we're going to cover all of that in the discussion that we have today. But just for the sake of clarity and even simplicity, let's start with the direct role. The evidence that suggests that ethanol is capable of causing insulin resistance directly. Now, there are some other, of course, indirect contributors that I think are very important and contribute to the overall effect of alcohol. When we consider someone who's drinking alcohol chronically or even somewhat infrequently, there's a lot more that goes into just the direct effect. It continues to have effects that actually that become broader. Seeler et al. In 2000 published a paper finding that ethanol directly inhibited the actions of the insulin receptor. So you'll recall from previous discussions, insulin being a peptide hormone, it's not capable of going into the cell itself. So if insulin wants to tell the cell to do something, it has to bind to a receptor to do that. And then once the receptor is bound with insulin or activated, it's going to elicit a series of events, ultimately leading to the cell responding and doing whatever insulin wanted it to do. Well, again, this paper by Seeler et al. In 2000 finds that ethanol was capable of disrupting the signal right at the origin, so directly blocking some of the effects at what's called the insulin receptor substrate, IRS, or specifically IRS1. So ethanol, if you incubate cells with ethanol or give them a little ethanol and then put some insulin into the culture, immediately you detect a reduced responsiveness. Also, ethanol, a paper by Shelmit et al. In 1988, finds that ethanol rapidly increases oxidative stress products, or these reactive oxygen species. And with more oxidative stress comes a shift in some of the phosphorylation state or the activation of some of those same enzymes in that insulin pathway or the insulin signaling cascade. So when insulin, again, comes to the cell and tells the cell to do something, as a part of that something is going to be a series of events, one enzyme affecting another, and then the change in oxidative stress with ethanol starts to disrupt some of this. And then another paper, Onishi et al., 2003, finds that this series of effects can go all the way to affecting GLUT4. GLUT4 is the glucose transporter. That is what's abundant in the most abundant tissues, skeletal muscle and fat. So when insulin comes to skeletal muscle and fat, knocks on the door of the cell via the insulin receptor, one of those cascades of events coming from the insulin coming and knocking, a lot of things start to happen. One of those things that starts to happen, one pathway of biochemical signaling, will result in GLUT4, this glucose transporter coming from within the cell, kind of opening up another door on the surface of the cell, allowing glucose to come in. Well, the paper by Onishi et al. again, 2003, finds that ethanol incubation disrupts that. And that would just be further evidence of insulin resistance caused by ethanol. Always important, particularly for me as a kind of cell biologist / physiologist, I want to go beyond the cell and then say, okay, but does the effect actually translate to a whole body effect? Because if it doesn't, then I would argue it becomes a little less important. However, the ethanol-induced insulin resistance that we see at the level of the cell in the 3 instances I just mentioned is indeed reflected at the level of the whole body. And I was able to find some very compelling evidence in humans using oral glucose tolerance tests. So there's a couple papers, Nagoro et al. 2005 and Metz 1969. So that's an older one, but that one actually is the one I like the most, which I'll share in just a moment. But in both of these instances, they had healthy human subjects drink the glucose drink like normal, and then track their glucose and their insulin response to this oral glucose load. In the first paper, Nagoro in 2005, these are healthy people. These are not alcoholics. They're not diabetics. When they drank ethanol before they had their OGTT, it resulted in this much, much higher response at 30 minutes with insulin in particular. So the insulin response was significantly higher. In fact, roughly about 50%. Interestingly, of the multiple studies I looked at in preparation for this discussion, including the Metz et al. Paper, also finds that insulin response to glucose is about 50% higher in across multiple studies. This does suggest a reduced sensitivity to the insulin, that the insulin is not working as well in response to this glucose load, and the body needs, in this case, substantially more. A 50% increase in insulin in order to clear the same amount of glucose does suggest a pretty profound metabolic shift that we're seeing, all because we've just added a little ethanol into the system. And then remember, these are humans, whole living humans. This is not at the level of the cell. So both Nagoro and Metz and others find that with any given amount of glucose, the body needs about 50% more insulin in order to get the same response. Now, some of you may be thinking that that we want to get a little more specific here, which I'm sympathetic to as a scientist, as a basic scientist or a cell biologist who studies the mechanisms of insulin resistance. I want to go all the way through this whole pathway from beginning to end. In every one of these instances, there are multiple papers finding that there is a common lipotoxic intermediate. Now, please refer to my earlier metabolic classroom entitled Energy Toxicity, where I sort of dispel the general myth that there's an inherent pathogenicity to energy or calories or fat, but rather it's a particular type of lipid, the sphingolipid ceramides. I very much am an advocate of the idea that overwhelmingly, perhaps not always, as a scientist, I want to be careful using that word, ceramides are a key intracellular cause of insulin resistance. And even with ethanol, that's the case. This has been shown in abundant, particularly animal studies where you can just pull out more tissues. You can get muscle from the animal, you can get the liver from the animal, you can get the brain from the animal, you can get the heart from the animal. You can't get human volunteers who are going to let you take a brain sample or a heart sample. Even a liver sample will be difficult to get from a human. But in these rodent studies, they've found that with ethanol treatment or consumption, ceramides is up in all of these tissues. It's up in the liver, it's up in the brain, it's up in the heart, resulting in increasing ceramides in all of these tissues, resulting in damage to those tissues, and certainly an insulin resistance effect. Ethanol is capable of directly causing insulin resistance at the level of the cell and at the level of the whole body in human studies. There are multiple methods for this, or multiple mechanisms that cause, that intermediate, that act as intermediary here, causing the insulin resistance in response to the ethanol. And ceramides are likely an end part. That's the ceramides, as I've discussed previously, directly come in and prevent the phosphorylation or the activation of a critical enzyme called AKT in that insulin pathway. So that's the direct effect. Now let's move on to the indirect effects. And by now you are familiar with my affection for alliteration, sorry. And that, so of the three indirect effects, I want to talk about sugar, sleep, and substrate. So these are the three S's of the indirect causes of insulin resistance in response to alcohol. All right, so the first one being sugar. The reality of many alcoholic beverages is that they're loaded with sugar. This is cocktails, beers, and some sweet wines. All of them contain either modest to very high amounts of sugar. Consider that in light of some of the human evidence I just mentioned, that when you combine glucose with ethanol, you are amplifying the effect. Ethanol alone isn't eliciting an insulin response. That's probably an important point I should have made. Ethanol alone is not inducing an insulin response, but it's not often consumed alone. It's often consumed with something else, whether it's the sugar in the drink or whether it's some of the starches in the nuts or whatever else the person may be eating while they're drinking that alcohol. But again, often sugar is present in some form. And this exaggerates the combination, amplifies the not only glycemic response, resulting in a significantly higher glucose level, but also a significantly higher insulin level. So insulin is working harder and ultimately failing, but all seeking to try to control the blood glucose response. And as you can imagine, as the individual continues to consume alcohol like this, day in, day out, that starts to contribute to a more chronic settling in of insulin resistance. Now let's move on to the next one. Sugar is an obvious one, but let me just again state it, that the excessive consumption of carbohydrates results in insulin resistance. Human evidence has shown that that high carb consumption can cause insulin resistance in just as little as 7 days. Also, if you have healthy insulin sensitive people eat just about 300 grams of carbs, which is not hard to do, their insulin is elevated for up to 10 to 12 hours. So it's easy to see how you can start stacking on these chronic insulin bumps, whereas one individual insulin spike isn't inherently pathogenic. It's when you continue to do this, which most people do, because most people consume a lot of refined starches and sugars, and they do so very often. Okay, so sugar is the first one of the indirect effects of alcohol causing insulin resistance. The next one is sleep. This one is surprising to most people because most people consider that alcohol consumption helps people sleep. What is true, and we'll cite a couple specific studies in a moment, is that alcohol increases or rather reduces the length of time it takes to fall asleep. In other words, people will fall asleep more quickly. But the impact of alcohol on sleep quality is that it disrupts sleep patterns and reduces overall sleep quality. And overwhelmingly, people will sleep poorly in response to alcohol. A poor night of sleep, this is its own topic of a metabolic classroom, is capable of causing insulin resistance. So let me just restate that. Even one bad night of sleep will cause demonstrable, obvious, quantifiable insulin resistance in humans the next day. Now, thankfully, one bad night can be corrected by one good night, but as you can imagine, as you continue to compound one bad night after another, the insulin resistance is a little stickier. One good night isn't going to be enough to wipe it out. And that is overwhelmingly a result of the stress hormones, particularly cortisol. So one bad night of sleep increases cortisol the next day, and cortisol will cause insulin resistance rather quite directly. And as you know from previous metabolic classrooms, cortisol is one of the primary causes of insulin resistance. Independent of any other stimulus, cortisol can cause insulin resistance, and just as a point of interest, does so by increasing ceramides within the cells of the body. Okay, now let's talk about a couple studies. The first one is Thakkar, Thakkar, 2015. It was published in the journal Alcohol. They found that these scientists found that in healthy non-alcoholics, acute alcohol, so just taking a little alcohol, decreased sleep latency. So that's just a fancy way of saying that the people did fall asleep faster than otherwise. However, While they did fall asleep faster than normal, the second half of the night was a much worse sleep. They had much less REM sleep, and they had much more limb movement. So they were just more agitated and moving their limbs around more, like with the restless leg syndrome, although they use a more technical term for it. But they were much more restless. So they had a significantly worse sleep in the second half. And then they note that with chronic alcohol drinkers or alcoholics, although that's a maybe too bold of a term, chronic alcohol drinkers, whether we would call them alcoholics or not, do experience severe sleep disruption, including an eventual chronic insomnia, which leads them to feel much sleepier during the day. A second study of many studies that I could show, and indeed there are so many that there are large reviews written about this, including this one by He et al., 2019. That's in the journal Current Opinion in Psychology, and they note that alcohol disrupts sleep architecture, which is a fancy way of just saying the normal structure of the sleep, and triggers substantial insomnia and disrupts circadian rhythm, once again disrupting sleep and causing daytime sleepiness. Now, the third of the indirect effects here is substrate. And you may be wondering why I'm using that word and how I'm using it. So to be a little clearer, what I mean by this is substrate competition. So calorie use, essentially, or nutrient use. So there are the main fuels in the body, the main fuels being glucose and fats. And then we have some sort of secondary fuels, although some may be primary fuels, like ketones, for example. Most people have very infrequent elevated ketones, so we will call ketones a less frequent or secondary fuel. Same with lactate. Lactate is a fuel that ought to be a topic of its own metabolic classroom. But these are the general fuels of the body, and they're all competing to get burned. When alcohol comes in, it kind of demands priority boarding. It's that obnoxious passenger who cuts right to the front of the line. And so everything behind has to wait in order to be burned. And so the glucose levels start to climb, the fat levels start to climb in the body, all because if ethanol has come in the system, it has demanded priority boarding on this metabolic bus. What this then manifests as, and this is largely why ethanol results in such heavy fat accumulation within the liver. So here we have this lipid accrual. Now, what you're thinking, those of you who tuned in to the previous energy toxicity lecture, is you're thinking, well, just storing more fat isn't a cause of insulin resistance. If you're thinking that, you're absolutely right. You're absolutely right. There are abundant studies to show that just having a tissue store more triglycerides, which is the form of fat storage, does not cause insulin resistance, even in the liver. So fatty liver isn't a cause of insulin resistance. There are distinct experiments to show this and prove it, in my mind, beyond any doubt. The same would go here. Now you're then wondering, well, Dr. Bikman, why are you even bringing it up then? Because there is still, if you do have an accumulation of lipid combined or fatty acids combined with inflammatory markers, which we're going to get to in just a second, then you do have a scenario, a metabolic milieu that is going to be stimulating ceramide accrual. And then we get back to, rather than the idea of energy toxicity, which I generally reject, we do come to this idea of lipotoxicity, where there is a very highly bioactive lipid, particularly the sphingolipid ceramides, rather than the inert glycerolipid triglycerides. So triglycerides are not a cause of insulin resistance. However, ceramides are. So this whole idea of substrate competition does explain why the body is getting loaded with higher glucose, higher fat stored in tissues like the liver, which is the main tissue that's metabolizing the ethanol in the first place. This starts to explain some of that. But remember also, we have the high insulin, and my lab has published previously that with this substrate competition idea in mind, the ethanol is demanding priority boarding. The carbs are going to start to rise. The insulin rises, and an increase in insulin, high elevated insulin, does stimulate ceramide biosynthesis, causing insulin resistance. So even though fatty liver disease is linked with insulin resistance, I am not one to say that it causes the insulin resistance. It's just that the overall metabolic scenario that's causing the fatty liver disease is also causing insulin resistance. Now, before we finish, I'd mentioned inflammation just a moment ago, and I wanted to end with that. And this would be, once again, a bit of an indirect cause, but it's not one that I didn't know whether I sort of consider it substantial enough to put it in with those first three: the sugar, sleep, and substrate. But in this case, it's the junctions of the intestinal epithelium. I've talked about this before, and I will talk about it much more in the future because it is relevant. You've all heard, of course, of the idea of leaky gut. Normally, the epithelium is a well-controlled layer of cells, and whatever is coming from the gut into the body is controlled by the cell itself. All of the epithelial cells, they'll recognize a molecule and say, I know you, you can come in. And it will pull it into itself on that front end and then release it into the body on the back end. So it's very controlled, moving through the cell itself. But similarly, the cell is able to look at something and say, you don't belong here, you can't come in. However, ethanol, where there's one cell, imagine another Ben right here by me, shoulder to shoulder, bumped in. That junction between the two of us is so tight. The junction between the two cells is so tight that nothing can get through. Ethanol starts to open that up. And now we start to have this paracellular transport. So the cell itself is saying, I recognize you and you can't come in. Oh, and you just slipped right by me. And so this control of substances is compromised. Again, in this case, all because of ethanol. As a point of interest, fructose does the same thing, albeit perhaps to a more modest degree. But what should be a very, very tight junction becomes leaky. And now things can get through. And one thing in particular that I have studied extensively, and you can look up work on this, just go to Google Scholar or PubMed and type in LPS and Bikman, and you'll find a couple good papers on the topic. But LPS is short for lipopolysaccharide. And these are bacterial, these are molecules that are on the surface of bacteria that are abundant in the gut. And while they are okay in the gut, they should not be in the blood. And as they move through this gap between the epithelial cells of the intestine, they get into the body and then cause this chronic inflammation or this systemic inflammation. And what my work found is that LPS will bind particular receptors on cells, telling the cell to increase its ceramide production. So here, once again, we have an indirect effect of alcohol causing insulin resistance, but through this leaky gut LPS absorption or invasion into the blood, LPS causing inflammation, then inflammatory pathways causing ceramide production, and then ceramides reaching a point where they begin to fight the insulin signal, resulting in insulin resistance. So this, if you will, would be the fourth of these indirect causes.
It's another supplement when just a good diet will do.